So it is my great pleasure to welcome Gesa Hartwigsen to our NeuroConnect seminar today. Gesa is a cognitive neuroscientist and psychologist leading the Cognition and Plasticity group at the Max Planck in Leipzig. Her work focuses on high-level cognitive functions, namely language, or also language. And in particular, she studies how brain networks react and adapt to events in life, like could be a brain lesion, but also aging. So just the dynamic morphing and adaptation of our brain network. So we are very excited to hear insights from her latest work. And I will leave you the floor. And I think she said she's okay with questions during the talk, if you want.
Yeah, thanks for having me. It's a great pleasure and my first time in Geneva, so I already enjoyed it a lot. And yeah, I think Valentina already said it all. So what I want to transfer to you is a bit about functional plasticity in the language network, so how our language system adapts to challenges. And in our group, we combine neuroimaging and neurostimulation to probe the system and then map the responses of the system. So the overarching question is how brain networks drive flexible adaptation to challenges during language processing. And as outlined here, challenges occur across the whole lifespan. Developing brains, as you all know, face challenges during learning. In our example, language learning is quite a challenge for the system. But even as young adult brains, when our brain is fully wired and there's only neural cell death, so everything goes down, we still need to adapt to challenges, for example, at train stations when there's a lot of noisy information that we need to suppress. Then aging brains are a very interesting model to study challenges and adaptation to challenges. And one of the big unresolved questions that I'm also very interested in is which changes in the connectivity of the language system as we age reflect compensatory efforts of the aging system to adjust to cognitive declines and hearing loss, and which of these changes may rather reflect the decline itself. And finally, at the end of the lifespan, and sometimes also earlier, we often face lesions to our brain, for example, after stroke or brain tumors. And it's a big challenge for the system to adjust to the challenge to such events and regain functions during language recovery. And I want to better understand how this works and which of the mechanisms of the brain, and especially of the language network interactions, help such relearning of language. So the overarching game is to uncover universal mechanisms of flexible adaptation during language processing. And I should mention here across the adult lifespan, because so far I'm not investigating language learning or development, at least not yet. So what do we know about the neural mechanisms of language processing in our brain? We know that for many years now, language is organized in distributed networks in our brain. So aside from Broca's area and Wernicke's area, previous meta-analysis have now demonstrated that language engages distributed networks, not just the dominant left hemisphere, also right hemispheric regions. I'm not so good with the pointer, so apologies for that. But also subcortical structures and the cerebellum. The cerebellum is a very interesting structure that has been neglected in language research for a long time, and I think it's now being revived. Now, when we study different language functions or subdomains, such as the processing of meaning of words, the semantics, or the processing of sound of words, the phonology, we can get more or less specialized maps. This is data from the largest meta-analysis so far that we've recently published, and it took us 5 years to gather all the informations from different studies, so quite an effort. And if you would now directly compare different subdomains, for example, semantics and phonology, you would get more or less specialized patterns of subnetworks that are engaged in these different tasks. So successful communication when we talk and listen to each other requires flexible interactions between such specialized subnetworks, which has been demonstrated in years of language research. Yes?
So I'm just trying to specialize, but your left semantics and your left phonology seems awfully similar to me.
Yes, this is because this is a general map here of different studies, different experiments that show activity at the less conservative level, that is all kinds of semantic tasks versus low-level baselines. So the specificity comes in when you do direct subtraction or direct comparison of these domains. So we also had that in our meta-analysis where we directly contrasted semantic tasks with phonological tasks. Then we got specialization. For example, the more anterior part of the inferior frontal gyrus was more distinguished for semantics and the posterior parts for phonology. But this is not shown at this overarching map. That's true, that looks more similar. So now one question is, what happens if it comes to a challenge? Where do we see changes in language-related activity and language-related interactions? And one way to address this is to combine neurostimulation with brain imaging, so with neuroimaging, fMRI or EEG, to challenge the system and then use this as a readout to map induced changes. We can also use, of course, behavioral studies, but then we don't know what's going on at the larger network level, at least if we stimulate over single areas. And a classic approach to do this is to apply neurostimulation, for example, TMS. We do a lot of TMS, plasticity-inducing TMS studies where we first stimulate an area and then map the induced changes at the network level, either with fMRI or with EEG for the neural signatures afterwards. And these plasticity-inducing brain stimulation protocols, they have been mainly established in the motor system, and they are assumed to be either inhibitory, so disrupt function, or facilitatory to improve function. And by applying them and mapping the consequences with fMRI, we can map so-called short-term reorganization or adaptive plasticity. That is the relatively short-term response of the network to a perturbation or to a facilitation. The effects of such protocols are thought to last probably 30 to 45 minutes. So that's the window where we can look into the relatively short-lived after effects of single sessions. Now we can also combine both approaches, EEG or fMRI, with neurostimulation in a simultaneous fashion to map the immediate responses at the network level. This becomes particularly interesting when you are interested in the immediate consequences of a perturbation or facilitation, or if you want to map something across the time course. For example, if you do a novel learning paradigm and you're interested in changes in task-related activity across the time course of the experiment and how this maps the stimulation-induced changes in learning. So both approaches allow to quantify the impact of neurostimulation at the neural network level. We can look at changes in task-related activity, task-related interactions, which we refer to as connectivity, and associate them with behavioral changes that are sometimes induced by neurostimulation to get a better understanding what happens if we target a specific area, for example, if we disrupt function, how do brain networks adapt to that, and how do they change their computations. And with this, we can build models that also help to better understand what happens after brain lesions such as stroke. And this is an example for stimulation-induced plasticity in the language network. So this picture is from a review where we summarized studies that had in common that they targeted language-related areas or auditory cognition areas with the aim to briefly interfere with function and then map the induced changes during fMRI afterwards. So you can see that most of these studies include classical language areas, the IFG or ATL, also auditory cortex. And one common observation of these very different studies that use different paradigms and tasks was that if they inhibited a specialized area, for example, the left IFG, for a short time with TMS and mapped the consequences afterwards, these studies revealed stronger engagement, so upregulation of homologous areas. What does this mean? After disruption of usually the left hemisphere, they found stronger activity in the contralateral homologous areas of the right hemisphere. And this is often interpreted in terms of compensatory activity increases after disruption of the dominant hemisphere, and such compensatory increases are thought to help the system to maintain function at a high level. With such studies, if you talk about compensation, it's always a bit of the question, what's the behavioral relevance of this? Because compensation more or less means the absence of a behavioral consequence. So this is always a bit tricky. And I think this is something we have to keep in mind for all neurostimulation studies. I will come back to that later. Yes. Yeah. So the question was, what's the timing here? So these studies usually work like this. You apply your brain stimulation protocol for up to 30 minutes, sometimes also just 40 seconds. And then you map the consequences within the last, let's say, next 30 to 45 minutes. So this is just a single session application where you map the relatively short-lived consequences. If you want to have more long-lasting effects, then you would have to use repeated sessions of stimulation, for example, for therapy. Now, based on these studies and many more, I had an idea for a model how flexible adaptation in the language network might work. And a key aspect that I want to focus on today of this model is that adaptation, so compensatory shifts in the activity, not only take place but within the specialized language network if I perturb a region, but also occur across areas. What does that mean? So let's assume we target a central hub in the language network and we try to disrupt it. Sometimes this is also called virtual lesions. I usually don't use this term anymore because it has nothing to do with the lesion, but yeah, that's a different story. So the idea is to perturb the activity and inhibit the system. What we observed in some of our studies is that this can induce strong inhibition at the whole network level, because if it's a hub region and it is connected with other areas in the network, then this is sometimes resulting in strong downregulation of the larger network, sometimes even including homologous areas. And now you would think this might have dramatic consequences at the behavioral level, namely that task performance is no longer possible. But what we observe, and this I put into the model, is that if the language system is decreased in its contribution, we see a stronger recruitment and upregulation of neighboring networks that are outside the language network, namely so-called domain general networks. What are domain general functions? With this, I refer to other higher cognitive functions such as attention, cognitive control, or working memory that are known to be support functions for all cognitive processes such as language or problem solving. So the idea is that if the language-specific network is downregulated by short-term perturbation, then stronger recruitment of domain general circuits helps to maintain function at a high level. I'm not suggesting that domain general networks can replace the language system. If this was the case, then we would not see the dramatic consequences of stroke-induced brain lesions. The idea is more that to some degree, they can compensate for the disruption. This is what we've learned from neurostimulation studies. And now one question that I want to walk you through in this talk is, if such recruitment of domain general networks or circuits is a universal mechanism that we not only observe in the young system if we challenge it with brain stimulation, but that we also see in aging brains and in lesioned brains after stroke. Because identifying such a mechanism is not only giving us insights into systems plasticity, but may also help to stimulate more efficient treatment options, for example, with non-invasive brain stimulation. Now, what do we know about flexible neural network recruitment in aging brains and the role of domain general networks here? This is work from a postdoc from my group, Sandra Martin, who's interested in neural reorganization in the aging language network. And she used for her PhD work a relatively simple task, namely verbal fluency, so semantic fluency. You could argue whether this is a language task at all or just an executive task, but let's assume semantic fluency is telling us something about semantic executive functions. So people just have to see categories and they have to name members of these categories during fMRI. And one observation that I want to walk you through is that older adults show increased facilitatory, so excitatory network interactions, which predicts their verbal fluency and in a positive way. So stronger network interactions are related to better verbal fluency performance. What did we see here in general? And this is something that I found across all our studies. If you compare old and young subjects, old subjects tend to have less activity in the language-related areas and also in many other functions. So overall, their activity is decreased. And some areas also showed positive activity, so more activity for older than young subjects, namely the right IFG here, and some central areas in the motor system and supplementary motor area. So more or less domain general areas that showed more activity in older than young subjects when they were performing a similar task. And I should say here that task performance was similar. So the task-related accuracy was not different, but old subjects are usually, they need longer to respond to the task. Now, what about age differences in connectivity? What you can see here is age differences in the underlying large-scale networks. So we used the task-related activity, extracted the underlying networks with an ICA approach, and then looked at the network interactions. And the general observation is that older brains, so old subjects, show increased task-related activity and interactions between domain general networks. So, and the interesting part is that older subjects mainly show positive network interactions. So these are the interactions shown in red here, for example, between the default mode networks or default mode network and attention-related networks. And these interactions are usually or often negative in young brains and switch to facilitatory positive interactions in old brain. So one observation is here, increased facilitatory network interactions, positive network interactions between so-called task positive and task negative networks. And now you could say, yeah, so what is that meaningful? What do task-related interactions tell us? And if you correlate that with semantic accuracy, then you can observe that the stronger facilitatory network interactions are associated with better fluency in older adults. In younger adults is the opposite. So strong network interactions between those interactions that are usually anti-correlated is associated with worse performance. For older subjects, it seems to be beneficial or helpful to maintain performance at a high level. So a first hint that there may be a change in the network interactions during fluency tasks towards more positive network interactions. To further investigate this, we used measures from graph theory. So graph theory gives you an idea how larger network interactions may work in the brain. And we used a measure called segregation. Segregation tells you how much networks form specialized communities. So how much networks talk within the network to each other, or if they are organized more globally. And here in this graph, you can't see much. It's just a nice illustration that the network architecture overall is different in old and young subjects. And now let's focus on this segregation measure. So we see less segregation in the old brains. So they have significantly less specialized communities, if you want to say so. So this is a second hint that there's something different in the old brain. So old brains show more between network interactions than young brains, and on the other side, less segregation, so less specialized communities. Yes? No, this is all task-related data. So during rest, such findings have already been reported, and we thought it's a bit unfortunate because no one knows if this is good or bad and how this associates with performance. Yeah, and then the final step, we were asking if old subjects show more of these between network interactions during a semantic task with high executive demands, can we probably improve task performance and increase these interactions with so-called facilitatory neurostimulation? So we used a protocol here called intermittent theta burst stimulation that is known from the motor system to increase corticospinal excitability. And one idea is that this may also improve semantic fluency. And we targeted the pre-SMA because this is an area of the semantic executive network at the intersection between semantics and executive functions that we also found strongly active in all our subjects. So this is a map from the old brains where they showed high activity. We used a very challenging semantic association task here where they had to associate pictures with features and only stimulated old subjects in this study. At the behavioral level, we did not find anything, but we found that old subjects had increased connectivity between these domain general networks after stimulation, and that the amount of change in connectivity, so the increase between the targeted area and an area of the dorsal attention network, the left superior parietal lobe, was associated with individual stimulation-induced efficiency. So the stronger the connectivity after TMS, the faster the stimulation-induced responses of the subjects. So this may indicate that facilitation of between network interactions, so more between network interactions after stimulations, are associated with better semantic efficiency, faster responses to challenging semantic tasks. So if we take all these findings together, yes, just a second, then this may indicate that old subjects have different strategies. These strategies include more recruitment of domain general areas. This is also seen in more between network interactions of networks that are usually anti-correlated in younger brains. And if we facilitate this, if we stimulate this with neurostimulation, this is associated with better or at least more efficient performance, which may point towards a compensatory strategy to compensate for the overall loss of executive functions. Yes?
I have a clarification question. So you said that there is more connectivity in older adults in the first place, right?
Yes.
And then there is more of it for everyone after stimulation?
Yeah, no.
Okay, just for older ones.
Yeah.
That's what I'm trying to ask about, I guess, how the performance, like, do we think, as traditionally, that younger adults perform better than older adults in unobstructed conditions?
Yeah.
Or is it the case that you chose semantic fluency because that's the one task where actually adults do maintain performance? All I'm trying to understand is that the take-home message should be that more distributed and more interaction is better for across the board, or only in the specific case where you've taken a subject and stimulated.
Yes. So as always with neurostimulation, excellent question. The picture is more complicated. So the first data were from the fluency task, so everything about fMRI. And there the picture was like this, the accuracy was not significantly different. So old subjects, we had high performing old subjects, they were still as accurate as young subjects, but less efficient, so they just take longer. And this is something that we often observe. And here in the neurostimulation studies, we just included a group of old subjects and they were their own controls. So we had a condition of placebo stimulation versus effective stimulation. So we don't know if young subjects would benefit from the stimulation, and I would doubt so, because performance-wise, even the old subjects did a pretty good job, so they might be at ceiling. And I think the only effect, initially we had hoped that we could also have a direct stimulation effect. This was not seen. The only thing that we found was we see increases in the connectivity between these domain general networks after stimulation and a decrease in the response speed. And these two things are correlated. So the more connectivity I have, the faster I get through the neurostimulation. So a bit of a complicated picture.
Yes. Quick follow-up. I was trying to understand the direction of the effect. Are you, by stimulation, pushing the older brain towards more older brain-likeness, with less segregation?
That is a very good question. So if you couldn't hear it, the question is, what is going on here? Are we pushing the brain to a state that is probably even more reflecting the old state? I think it's more complicated than that. So the segregation measure, the general segregation measure, is a whole brain measure. And here we see, we did a whole brain analysis, but only found a very specific change in the connectivity between the targeted area and one other node from a domain general network. So we didn't see an overall change in the large whole brain segregation. So I think what we are probably optimizing or pushing a bit is the specific interaction between a node that is known to be super important for semantic executive functions and another node that may be required for attention. Yeah. Yes.
Two clarification questions. So to measure connectivity, you did the stimulation at the same time as you were doing MRI, or you doing connectivity with EEG, or were you doing...
Yeah, so the question was, is this a simultaneous measurement? And no, it's not. So it's a plasticity inducing protocol. Again, we first stimulate and then afterwards measure the connectivity during the task during fMRI. So it's an after effect of the stimulation. So the brain had a short time to respond to the stimulation. Yes.
And then it seems that in your connectivity between the pre-estimate and the left SPL, actually, if you look at the 0 line, I don't know, maybe these are Z-score?
Yeah, I think these are normalized data because it's the delta in the connectivity between the active stimulation and the placebo condition in individual subjects.
So you did Z-score, so you know that if you were looking separately active versus sham, you have actually greater connectivity.
Yeah.
It's not that you have...
No, that these are not reversed effects, yeah, because we Z-scored the data. Yes, so this was also the most, I think, complicated part of my talk. I promise now it's getting a bit easier and probably a bit more intuitive. And I want to move from the aging brain, where we have hints now that domain general connectivity may be helpful for efficient performance, at least in some cases. We move to something that I got very interested in, and that is dyslexia, a learning disorders, where it has been discussed that maybe domain general networks or aberrant domain general network contribution also has a role in this disorder. So this is work by Sabrina Turka, who was a postdoc in my group and whom some of you might know, because I think she also has a connection and works with Nali Golestani now. So what we wanted to do is better understand what happens when dyslexic individuals have to read. Can we characterize this as a network disorder? And when you compare reading performance and reading-related activity in adults with dyslexia versus controls, then what is striking, and this is from a study where adults with dyslexia had to read complex pseudo words, so words without any meanings with 3 and 4 syllables, a very challenging task for them. We see less activity in these adults with dyslexia relative to controls, especially in reading-related areas. So these are typical left hemispheric reading areas, supramarginal gyrus, left ventral occipitotemporal cortex, but also right hemispheric areas, so the so-called extending reading network, right VOTC and the right cerebellum. So the first observation is massively reduced activity during the same task, so-called hypoactivity, less activity. This has been reported in many studies. Now, the novel thing that we investigated was how do these networks that show less activity during pseudo-word reading, how are they connected? How do they interact during reading? And here we found that it's not just massive hypoactivity, but also massive hypoconnectivity. So we used all these seeds that had been shown to be underactive in these subjects and used them as seed areas for psychophysiological interactions during fMRI and looked at changes in task-related interactions. And the observation was that the reading performance is not only characterized by underactivation in people with dyslexia, but also massive hypoconnectivity. And what I illustrate here is the seed region in the right cerebellum. You can see that it's relative to normal readers. There's, during word and pseudo-word reading, massive hypoconnectivity to the whole brain. So massively reduced interaction with the whole brain relative to normal readers. And in further analysis, we showed that these hypoconnectivity patterns are also associated with their individual impairments as measured by reading and working memory tasks. So it seems that yes, we can characterize dyslexia as a network disorder. It's more than just low activity in reading-related areas. The interaction between those areas and the extended reading network, also including right hemispheric areas, especially the cerebellum, seems to play an important role here. And we've also done some neurostimulation studies here. I just brought a brief example because the results were super complex. And we targeted the left temporoparietal cortex. This is a core area of the reading network. And again, with this intermittent theta burst stimulation. And we found a slight but significant improvement after a single session of TMS over the temporoparietal cortex in subjects with dyslexia. This is now, again, only subjects with dyslexia against the placebo condition. And as a side note, what you can do if you target something with TMS, you can simulate the induced electrical field. And here, more reddish or pinkish means that the current induced is stronger to make sure that you hit the right area. And this is something that I'm more and more doing to improve our brain stimulation accuracy. So it seems that facilitatory stimulation over a reading-related node improves reading performance at least slightly. And what we also found, what I didn't show here, is that increases network interactions in dyslexia. So it seems here that dyslexia is a condition which may be characterized by massive hypoactivity, hypoconnectivity, and if we facilitate the network, then we can shift these network interactions a bit more to the state that we observe in normal readers. Now, moving to the last part is what do we know about flexible neural network recruitment in lesioned brains and the role of domain general networks in language recovery. And I'm very interested in that because for a long time, everyone thought that in aphasia recovery after stroke, it's mainly the left hemispheric language areas that are not damaged, which play a role in recovery. Then there were some studies by the Richard Weiss group that emphasized also domain general circuits, so attention, cognitive control, right hemispheric areas seem to play a role. And we got interested in that to understand the specific role of domain general networks or regions in language recovery. This is data from Dorothee Sauer's group in Leipzig, a study led by Annika Stockert, a postdoc in her lab, where we were interested in longitudinal language recovery. And this is a very unique data set that has been collected for many years. I was only part of it for a short time, where people with aphasia are recruited 3 times across the time course of their recovery. So they are first recruited in the acute phase after stroke, in the first days after stroke, then in the subacute phase in the first 1 to 2 weeks after stroke, and then again in the chronic stage at least 6 months after stroke. And the idea is that this longitudinal design allows you to investigate how changes in task-related activity occur and how they may be related to language recovery. And the paradigm you can imagine for acute stroke patients, immediately after stroke, you have to use a very simple language paradigm. So they just listen to sentences, and this activation was compared with listening to reversed speech. So just a passive paradigm. And if you look at changes in task-related activity across the time course of recovery, I only selected one very interesting finding here. And this is for a subgroup of patients who had lesions in the temporal-parietal cortex. So temporal-parietal stroke, we also had a group with frontal lesions. And these are 17 patients who are measured repeatedly. In the acute phase, directly after stroke, you see a massive reduction in overall activity when you compare them with controls. And what you can see here is the subacute phase. So in the first weeks after stroke, when these patients listen to speech versus reversed speech, this part here is masked out because there were the lesions. You see an increase in task-related activity in bilateral frontal and prefrontal regions, including classical domain general areas like the insular cortex, the left and right dorsolateral prefrontal cortex, so these classical working memory areas, and also these midline structures here, the SMA again, and underlying dorsal anterior cingulate cortex. So it seems that already from the acute phase to the subacute phase, there is a massive increase in activity. This is interesting and has been shown before. And we could also link that to behavioral performance improvements. So this is a language comprehension score taken from the Aachen Aphasia Battery, a standard test diagnostic of aphasia. And we can see that from the acute to the subacute phase, there's an increase in language performance. This is normal recovery. This is good. And the stronger the increase in performance is associated with stronger increase in task-related activity in these areas from the acute to the subacute phase. So at least in patients with temporal lesions, we have a hint that early upregulation of domain general frontal and prefrontal regions is associated with better language recovery. I didn't bring the data for the frontal group. The patterns look slightly different. So it's interesting to keep in mind that this may be lesion specific. Now, in the next study, this is a PhD student from my group, Zhezhao Yang, who thus investigated the underlying changes in task-related connectivity. We now look at the full group of subjects, so it's the same data, and now instead of activity, we investigate connectivity. So again, acute, subacute, and chronic phase longitudinal design, patients with either temporal-parietal lesions or frontal lesions. And we extracted data, seed regions, based on those patterns that showed the strongest increase in activity across the time course of recovery. So we had 2 perilesional language areas, so activity in the left IFG and posterior temporal cortex. We made sure that this was outside the lesion in each participant. We included the right IFG because its role in language recovery is still hotly debated. We have some ideas that it may play a beneficial role. I won't talk about this today, unfortunately. And then we had 3 domain general areas, left and right DLPFC, and this SMA area again, because these were the areas where we showed the strongest overall increase in task-related activity. And now what we did was to use an effective connectivity measure called dynamic causal modeling, where you can model how across the time course of recovery we see changes in task-related interactions. And this method claims that you can also investigate the direction, so which region is driving connectivity in which other areas, and whether it's facilitatory or inhibitory. We can criticize the model, but if we trust the output, we can here see what happens already in the acute phase. So in the very first days after stroke, the activity in the patients is massively reduced, but we see relatively strong connectivity between the domain general areas and the language-related areas. So it seems that already in the acute phase, in the first days after stroke, domain general areas strongly influence the remaining language areas, and this influence is positive, so it's facilitatory. And we also associated this increase in the connectivity with language performance, so how well they performed in the acute phase, and with language improvement. So when we first look at language performance, then it's interesting to note that it seems that especially those patients with temporoparietal lesions, so posterior lesions, benefit from increased connectivity. So only in the acute phase, we see a strong association with domain general interaction to language areas and better language performance. So early increase in connectivity is associated with good preserved performance, and maybe more important because all the clinicians always want to know, is this also associated with later language improvements, so how well the patients recover from the stroke? And yes, it seems to be the case, and even across groups. So early increase in the connectivity from domain general areas on the temporal cortex is overall in all patients associated with later language improvements in the subacute phase and in the chronic phase. So I think this is the first evidence that a very early impact of the domain general regions on the temporal cortex is actually good for recovery. And one idea that could now be followed up upon is to use neurostimulation, facilitatory neurostimulation early on after stroke, and not only target the language areas or the right prefrontal cortex, but also use these domain general areas as target candidates to facilitate recovery. And I think a good candidate area would be the pre-SMA, because it was one of the areas where we showed very consistent findings in activity changes and connectivity changes. It also seems to play a role in the aging network. And my idea is that it's probably not so much the semantic related content, but more its domain general role. And finally, what we're currently doing, and I apologize for the bit of a messy slide, is to better understand if we can identify predictors of good language recovery with, by putting into a model, and this is a representational similarity analysis, which takes into account at the same time very different predictors. And I will walk you through this because it could be interesting. So we have now from our 2 studies the finding that activity changes across the time course of recovery and connectivity changes. So we thought if we put all in one model and identify the predictors that significantly contribute to language ability and language improvement, maybe we learn more about the brain aside from very specific network interactions. So as predictors, we had language performance during each phase. So in the acute, subacute, and chronic phase, we had activations in the acute and subacute phase, and connectivity in the early phases, plus lesions and age. And we use these predictors to predict how language recovers in the chronic phase, and what if these predictors can tell us anything about especially language improvement. And something that is not surprising is that language performance is, of course, always a good predictor of language ability. So language performance is highly correlated across all phases. Lesions are also a well-known predictor of actual performance and later outcome. This is also probably not so interesting because it's quite obvious. If you have a large lesion, then the chance that you have worse performance is super high. Now, what I found more interesting here is the connectivity. And everything that has a star here is significant. And what you can see here is that we repeatedly find that connectivity here now means across all seed areas that we threw into the analysis, that increased connectivity in the subacute phase is a relatively good predictor. And I think for an RSA analysis, these values that don't look impressive are okayish, are a good predictor of performance already early on and later language improvement. So subacute connectivity across all nodes, we don't know where it comes from, if it's a specific connection or all the connections between language-related areas and domain general and language areas. An increase in these connectivity parameters is associated with preserved early performance and early until late language improvement. So I think this is a last piece of information that was needed to really make a claim that domain general networks and the connectivity and changes in the connectivity between language and domain general networks are also associated with preserved language performance after stroke and especially with later language recovery after stroke. And this brings me to the summary. So what does this tell us about flexible adaptation during language processing? I guess everyone agrees that neural networks remain flexible across the adult lifespan. One idea is that flexibility does not only mean within the language circuit, but also flexible adaptation during language processing is needed. And we have some ideas now that domain general networks here make a core contribution. One question that remains a bit open, I think the strongest evidence that we have so far is from our lesion data, but especially from our neurostimulation work, an open question is, what is the behavioral relevance of compensatory changes? So if I disrupt one network and then I see a bit more activity or connectivity of a different network, is this really meaningful? To really probe this, we would need a network stimulation approach where we simultaneously target language and domain general areas and see if this is really functionally relevant. And if domain general network compensation is really meaningful, then if we additionally target these areas and disrupt them, we should see a stronger performance impairment. And this is something that we are currently working on. And another line of thought for future research that we're currently exploring is modeling approaches to use large language models to better understand our stimulation effects, because stimulation effects often don't work the way you want them to work, and to have a better systems understanding of such effects. And now in principle, I could stop for discussion or show you an outlook of some random thoughts, but maybe we take questions first. So thanks for listening and looking forward to the questions now.